My name is Dr. Kristi Baker, and I'm an associate professor at the University of Alberta in the Department of Cancer Biology and Medical Microbiology and Immunology. So my lab is really focused on looking at the kind of dialogue between the immune system and colon cancer. And we're really kind of focused on studying different types of colon cancer and how they act or interact with the immune system. There are some types that respond very well to immune-based therapies, and that's only a small percent though. That's only about 15%. And the other kind of 85% of colon cancers really don't respond well to immune therapies. And we're really focused on studying that small 15% of cancers to learn what is it about them that allows them to be responsive to immune-based therapies, in the hopes that we'll be able to learn mechanisms that we can then use to make therapies that will help improve the lives of patients with that other 85% of colon cancers. I really wanted to do work that was going to be as applicable to patients as possible. And in order to do that, we really wanted to use the best model systems. So one of the things that we set up when we first started the lab was to get an orthotopic colon cancer system going in our animal models. And basically what that is a lot of the time when you do tumor research, you inject the tumor into the back of the mouse because it's accessible, it's measurable, and it's not very invasive for the animal, so it's good ethically. But that doesn't put the tumor in the normal environment where it's typically found. And so what we developed was a model where we could actually take engineered colon cancer cells that we were able to genetically alter in the lab, and actually then implant these directly into the wall of the intestine. So we have a little colonoscopy machine that we use to inject our tumors into the mice. And then we'll watch them grow in a non-invasive way. And we're actually able to see the tumor growth over time, and then to look at actually how it's dialoguing with the actual environment in which colon cancer would normally be growing. I think the second thing that really sets us apart is that we use a lot of human models, ex vivo models. We have been collaborating with some surgeons here to get samples of patient tumors. They're removed kind of during normal patient treatment, just as part of the resection of the tumor. And what we've been able to do is set up organoid cultures. And what those are basically a way to be able to grow patient cancer cells long-term, over time, in an environment that allows them to kind of maintain the biology that they have in the patient. And it allows them to maintain a lot of these cell-cell interactions that are needed to kind of maintain that normal biology. And then using this technique, we're actually able to perform co-cultures with different types of immune cells that we get from the patient's blood, or that we purify from the patient's tumor at the same time. And so I think having those two model systems were really kind of really at the forefront of the bench to bedside transition, where we're doing the basic science research, but we're positioning it so that it will be easily translatable or very highly translatable into patients in the hopefully not too distant future. We do a lot of gene expression, we do a lot of sequencing, and I think that one of the things that I really truly appreciate about the Applied Biosystems technologies is that they're really robust platforms, but they're also really accessible platforms. I really do appreciate that, both the development that goes into these products and also the way that they're made accessible to the researchers who are using them. One of the things that we use most is the Quant Studio qPCR system. It allows us to do a lot of multiplex analysis and look at really a lot of different targets at one time in a really robust way. And that allows us to use our precious samples, so the samples that we get from patients and the samples that we get even from our animal experiments. It allows us to extract as much data as possible in a really reliable manner, as opposed to having to go back and consume more sample or recruit more patients. It's hard to choose one favorite part about being a scientist. I really love the chance that we get to ask questions that a lot of people have asked before, sometimes they're questions that nobody's asked before. But whereas a lot of people kind of ask the question and ponder about it and move on, we have the tools and the chance to actually be able to probe some of these questions and to really give a good shot at answering them. And I love that challenge.